Good afternoon. I'm Mary Jerome, Director of Medical Communication and Education at the MMRF, and I'd like to welcome all of you to our Multiple Myeloma Patient Symposium here in Boston. Thanks so much for coming. All of us at the MMRF have been here in Boston for the past few days, attending one of the premier meetings specific to myeloma, the International Myeloma Workshop. This meeting is held every other year, and it brings together renowned myeloma specialists from all over the world who present and discuss the latest advances in myeloma clinical research. We're very fortunate to be able to bring you updates from this important meeting and to answer your questions throughout the program. And today, we're also broadcasting this meeting live, so I'd like to welcome everyone watching us from home. And today, we are joined by a number of esteemed colleagues and myeloma faculty who you'll hear from throughout the afternoon. We'd like to thank Adaptive Biotechnologies, Amgen, Celgene, Cure Magazine, Karyopharm, Oncopeptides, Sanofi Genzyme, and Takeda Oncology for supporting this symposium. Before we begin, I've got a few housekeeping notes here. You'll be able to fully participate in today's symposium through the iPads that are located at your seats and through your computers for those of you who are watching remotely. You'll be able to follow along with the presentations, answer audience response questions, take notes, and submit questions directly to the panel of speakers. Also on the iPad is our activity evaluation. We ask that you complete the evaluation at the end of the symposium because your feedback is really very important for us for the planning of future educational activities. At the conclusion of the symposium, please be sure to leave your iPads at your seats. I know everybody likes to take them home, but you can't have them. And the rebroadcast of the live webcast portion of this event will be up at a later date. Please tune in to our website and you'll be able to watch it all over again. And I'd like to ask everyone to turn off their cell phones at this time so we don't have any extraneous noises for those who are watching at home. Also, for anyone who parked using the valet at the hotel, if you haven't already done so, make sure that you get a sticker from our registration table outside to validate your valet parking so that we can make sure that you don't have to pay more than you should. So today's faculty, we have Irene Gabriel from the Dana-Farber Cancer Institute, Maria Victoria Matios from the University of Hospital Salamanca in Spain, Jesus San Miguel from the University of Navarra in Spain, and Paul Richardson from the Dana-Farber Cancer Institute. Ann Quinn Young, who is the MMRF Chief Marketing and Development Officer of the MMRF, will be moderating our discussions today. So here's the agenda for today's meeting. There'll be plenty of opportunities for moderated discussion times on the agenda. We're looking forward to a spirited discussion. And remember that you can submit questions to the speakers using your iPads at any time during the symposium. And for those of you watching at home, you can also submit questions during the live webcast. We also have several supporters here who have tables in the rear of our meeting room. They're here to support you and answer any questions you might have, so feel free to drop by their tables and visit them immediately after the symposium. We'll have refreshments available after the symposium at this time. Okay, so I'm going to get you guys all warmed up with your iPads with these audience response questions. So let's start off with a few questions. The first question, are you a patient, a caregiver, or other? Okay, so it looks like we have mostly patients and caregivers, a few of the folks in the other category. That's great. The next question, what stage is your myeloma? And if you're a caregiver, what is the stage of the patient's myeloma? Newly diagnosed, relapsed refractory, in remission, still on therapy, in remission, not on therapy, MGUS or smoldering myeloma, not currently requiring treatment, other, or I don't know. Okay, so it looks like most of our folks are in remission and still on therapy. Next question. Have you had a stem cell transplant? No, but I will soon. No, but I'm considering one. No, my doctor tells me I'm not a candidate. Or yes. Okay, looks like we have a majority of transplanted folks here. Do you know if you've had any molecular characterization performed on your myeloma cells, such as FISH, cytogenetics, or sequencing? So you can see the choices there. Either no, or you had FISH, you had cytogenetics, you had sequencing, a multiple of tests performed, or I don't know. Okay, we've got a pretty good spread here. Not many people have had sequencing. Okay. So is this your first time participating in an MMRF event? Yes or no? Okay, about half and half. So we're happy. Those of you we haven't seen before, welcome. We're happy to have you. And those of you who are returning, we're happy to see you again. So how familiar were you with the work that the MMRF does before today? Not familiar, somewhat familiar, or familiar? Okay, good. It looks like you folks are fairly familiar with what we're doing, which is fantastic. And it looks like I've finally asked all the questions that I have. So on that note, I will now introduce Ann Quinn Young, who is the Chief Marketing and Development Officer for the MMRF. Ann?
Well, welcome everyone. This is an amazing turnout for a Sunday afternoon. I'd also like to thank our faculty because they have been here for 4 days. There have been 600 abstracts focused solely on multiple myeloma. There were 3,000 people from 41 countries. It was absolutely incredible. The progress that we're seeing is truly unprecedented in any other cancer. And it is such a gift for us to be able to be here today and give you the information hot off the presses. As Mary mentioned, we have our friends from Dana-Farber, who you know well, Irene, Gabriel, and Paul Richardson, but we're also so thrilled to have international faculty here as well, Jesus San Miguel and Mary V. Mateos from Spain. For a lot of these symposia, and again, half of you, it sounds like, have been to some before from the MMRF, it's usually limited to faculty within the U.S. because of travel and whatnot. So this is really such a joy. So thank you all for being here. I know we started a little bit late, so I'll try to run through this pretty quickly, but especially because half of you really are new to the MMRF and about almost half said weren't quite familiar with some of the work we've done. I want to take you through this quickly. So again, to really emphasize it, our mission is to find a cure for each and every patient. And we work with the entire community to do that. And at the center of everything we do are the patients. And since most of you, or half of you, aren't familiar with our work, I encourage you, go to our YouTube channel. There's a video that starts playing as soon as you go on there with 3 pretty well-known patients, Tom Brokaw, Kathy, our founder, who you just heard from, and Dick Parsons. And they talk really eloquently about what the work the MMRF does and what it means for all patients. Oh. So, as I mentioned, the MMRF works with the entire community. We sit at the center of patients and caregivers, all the companies, and you see we have an amazing assortment of them here today, not only supporting really important programs like this, but helping us advance the most promising treatments for patients. We work with the leading academic centers, community centers. You'll hear in a little bit, we're now working with venture capital firms and data and technology companies as well. Because this is a complicated disease. We've been at this a long time. This community is so committed, so tenacious, but it's complicated. It's really heterogeneous. As you know, we've made tremendous progress in terms of new treatments and extending patients' lives, but we're not yet at a cure to every patient. So it really takes this entire community coming together. Oh, okay. Again, it's been a community effort for the last 20 years. The MMRF prides itself on working really urgently. We're reminded every day, because Kathy is our founder, she's still working with us each and every day, that we're not quite to the finish line yet. So from the very beginning, our focus was, let's build an organization that's focused solely on research that's accelerating new drugs for patients. And you know, we established a model where we would identify what's standing in our way of delivering new and better treatments to patients and what's needed to address that. So in the beginning, again, there was no funding, so we started the organization to jumpstart research. When we talked to researchers and there weren't enough biological samples, the tissue where your myeloma is, to understand the disease, we built a tissue bank. We then used that tissue to sequence the myeloma genome and find new targets for the disease. And we were committed from that time to putting all of that data into the public domain to make sure that researchers had access to it, that it wasn't sitting in an organization or a company, that everyone could have access to that. We built on that with our COMPASS study, where we now have the largest genomics data set of any cancer. And through the findings that we've identified from our original genomics work and through COMPASS, we launched the MyDrug study, which is a first platform study in myeloma where patients are profiled and assigned arms based on their genomic mutations. I mentioned our partners. Some of our very close partners are 25 centers that work with us on our clinical network. Together, we've opened 80 different clinical trials, more than 30 different agents. Many of those have become FDA approved. We started with 4 centers. Dana-Farber has been with us since day one, back in 2004, and today we're at 25 centers strong. And I use the MyDrug study as an example of a program that would not happen without the MMRF working with all of these centers. Don't worry about the complicated, the drug arms and the schema, but what the real take home here is, when you look, we sequence patients who have relapsed early off of stem cell transplant or their initial therapy. So these are patients at high risk of progression again. We profile them and then assign them to an arm based on their mutation, based on the specific characteristics of their disease. And most of these alterations happen in 10 to 20% of patients. So you need many different centers working together to be able to enroll each of these arms. So the MMRF is working with 17 of our centers across the MMRC to make this happen. On the far left, you can see for patients who don't have an actionable alteration, we have an arm for those patients as well. And we'll be adding arms as we find new alterations through our work, and also for patients who don't have the mutations. So what's before us today? Before us today are some new challenges, some old challenges. I won't go through this in detail, but if you have any questions about our latest strategic plan, what we call our Smarter, Faster, Cure plan, please see any of us, me, Mary, Alicia, Chris, there's a number of Daniel, a number of us in the audience who can take you through this a little bit more. But essentially there's 3, and recently we've added a 4th pillar. The first one is that immunotherapy is transforming outcomes for diseases like lung cancer, melanoma, and we've seen some amazing hints of promise in myeloma with CAR-T and some other, now some BiTEs, some really exciting data you'll hear at the meeting today, but we really haven't brought its full potential to myeloma. So we are very much focused on not only identifying new targets and standardizing some of the tests, but bringing those promising treatments forward. The second piece is that with all of these new drug approvals, and patients doing so well, there's a sense that there's not a pathway for companies to get FDA approval for their drugs. So we started what's called the Myeloma Investment Fund, where we're investing early in companies that have promising assets to make sure that they bring them to myeloma. And then we're still focused on building out data because it's a heterogeneous disease and we need enough data to help every patient understand what's the right treatment in the right sequence to extend lives and get to cures. And then finally, and you'll hear a little bit more about this from Irene, we're very much focused now on early disease. For all of those patients, and I know many of them in the room, are not newly diagnosed, believe me, our commitment is still to make sure that you always have the right treatment when you need it. But we now feel like we're at a point we can also start to figure out how do we delay progression either temporarily or forever for the future generations of patients. So to focus very briefly on the data pillar, and again, if you have questions, please see us after. We've built the largest genomics data set with Compass. We have a number of other data sets, and we're now pulling them together into one big data hub that we call the MMRF Cure Cloud. And the purpose for that is to identify new targets, identify biomarkers, and come up with eventually treat care pathways, but in the meantime, enough data for patients and their doctors to make data-driven decisions based on the unique characteristics of the patient's disease. What's really exciting is that how we're building out this big data hub. On October 1st, all of you will see the news, and some of you, I actually saw at least one person registered who was part of the pilot program for this, but we're opening up the opportunity for all patients to share their data into the Cure Cloud. We started a registry a little over a year ago. We have 80 patients in that registry. Those patients signed up online. They signed a medical waiver, told us where they were treated. We were able to pull their medical records. We sent a phlebotomist to their home, got a blood sample. We've sequenced it, and that'll be put together with all of the other patients, again, to try to figure out how do we identify the subtypes and figure out how best to treat each group of patients. So we did the pilot, and now we're opening it up to every patient on October 1st. So stay tuned, look at your email, look at social media. And then in the next 6 months or so, we'll be changing the program yet again. So in the first 6 months, it's just sharing data, advancing progress, being part of the cure. Next year, we will be using that same panel to sequence your blood, but we'll be able to give the results back to you and your doctor. So that if there's a specific mutation that could qualify you for my drug or any other trial or an off-label treatment, you'll know that. So that's information that you wouldn't have otherwise had. So stay tuned. I encourage everyone to sign up in this initial phase, and then next year is when it gets really exciting. So I won't spend very much time at all on these last couple slides, but I show this because this is quite honestly just a fraction of the drugs in the immune pipeline. This is managed by my colleague Chris Williams in the back. And it's amazing. There's so many promising assets coming forward, so much data that was presented at this meeting, but we need to figure out how best to use these drugs, these treatments, in combination for specific types of patients. So I'll end with this. I started with our mission has always been the same. From day one, Kathy didn't really dare to dream that there'd be a cure in her lifetime, but that's changing now. And we're here working as urgently as we can with all of our amazing partners to try to cure each and every patient. And we're going to do it with the plan, working smarter, working faster, and working toward a cure. So my final slide, and again, this is something, please talk to any of us at the MMRF about here or following the meeting. There's so many different ways for so many of you to get involved. I'm sure there's many here who, again, I know at least one person was in the Cure Cloud Registry pilot. I'm sure many of you have done our 5K Boston race. But again, there's an opportunity to share your data, to join the patient registry, to enroll in a clinical trial, connect with us across social media, and then we have an amazing array of events for you, for patients, for caregivers, for family members who can do something. Join a 5K, run a marathon, climb a mountain, as some of our patients and partners have done. But there's many ways. So the first step is being here, so I thank you for that. And now it's my pleasure to turn it over to Irene Gobriel, who helped put this program together and was also one of the now very tired co-chairs of the International Myeloma Workshop.
Thank you, Anne. Good afternoon, everyone. And it's really a pleasure and it's really an honor to see everyone here. Just like Anne said, I think you all empower us, patients, caregivers, family members. It is because of you and because of your involvement we are doing all the clinical trials and all the work we're doing, and we want your support and your help, active participation, telling us what you want, being part of the Cure Cloud, being part of clinical trials. This is the only way we can move forward and truly cure multiple myeloma. And we've learned how truly our patients empower us and empower themselves by being involved. So this is an exciting time for us, so thank you for being here and being part of this. I think we have today 4 different people showing you what we just learned in the last 3 days in the myeloma meeting, and it was a beautiful meeting with a lot of interactions. We actually have the president of the International Myeloma Society who's here with us, and it's wonderful also to have 2 of our European colleagues, the stars of our European colleagues here, Mariví Matios and Jesus Miguel, to show you the whole spectrum of how we work together nationally and internationally to bring together the best treatments for our patients with myeloma. So without further ado, I'll introduce Jesus and Mariví to present their data, and then it will be followed by Paul Richardson and myself. Thank you.
Thank you very much, Irene. It's for me a pleasure just to be here. I started my studies in medicine in 1970, and I did because I want to cure patients. This was the decision to go to medicine, to cure. I did my PhD thesis on multiple myeloma in 1980, and at that time was really an incurable disease. On Friday, I was invited just to give the opening lecture in this symposium about the possibility of curing myeloma. And I talk about several actions. One was to investigate the high-risk patients, just to offer them differential treatment with the new immunotherapeutic approaches, to offer the standard risk, the best possible approach, early detection, early intervention, and to use the best possible methods, techniques, to measure the efficacy of the treatment to eradicate the disease. And I think we are getting close. And it's a pleasure also to be here because in the MMRF signature is cure. And this is the goal of all the doctors. And without further delay, let me start just to share with you what has been presented in this meeting about prognostic factors, genetics, MRD, in order to try to guide treatment decisions. Unfortunately, there is an imbalance between the biological information that we have from all of you and how we use this information in treatment decision. In other words, frequently we treat uniformly many patients without separating the high and the standard risk, without taking into consideration how was the profound enough the response to the treatment. And I think this is going to change in the coming years. Regarding prognosis in myeloma is the same as the prognosis in all cancers, and is driven by 3 factors. One is the host. Age performance score influences the prognosis. Second is the tumor mass and organ damage. In myeloma, you have the CRAB symptoms that also influence the prognosis. But particularly important are the characteristics of the tumor clone. And within the characteristics of the tumor clone, there are 2 main players. One is the genetics of the tumor, and the second is the interplay between the tumor and the microenvironment. Regarding genetics, as I have already mentioned, have a clear influence in the prognosis, and until recently, we only check the genetic information by FIS, but now we are starting also to use genome sequencing. And this is important because we have learned in the last 10 years that some genetic abnormalities, such as translocation on 414, 1416, or particular deletion in 17p, influence the prognosis, and this has been confirmed in the gene expression profiling analysis. And it's not the same if you have one genetic abnormalities or if you have 2 or 3. The coexistence of several of these have a more impact on the outcome of the patients. And this has been also utilized in order to establish the international staging system. that include the information about the tumor load and also information about the genetics. And as you can see, the outcome in advanced stage revised ISS3 that include poor risk cytogenetics is clearly different from the outcome in a standard risk stage 1 or a stage 2 patient. Therefore, this information should drive the treatment in the near future. We should not offer the same treatment to all the patients. The COMPASS study, I think this is a landmark study, and we have benefited all over the world from this study that was therefore recruiting DNA and genetic information from United States, Canada, Italy, and Spain. And this informed you about the frequency of genetic abnormalities, the role of these genetic abnormalities in myeloma, and to allow you to do a better risk assessment. In this meeting, 3 interesting studies based on the COMPASS information that is available for all the community, then this is really a major advantage for all of us. 3 studies have been presented, and one of them illustrates the presence of a subtype of patients called PR that exhibit high-risk features independent of the clinical staging system that I have already mentioned. The second study was focusing on predictors of early relapse, and it has been shown in this study that in addition of the well-established mutation of P53 or high LDH levels, or even now amplification of chromosome 1, mutation in ILL5 is also very important because the outcome of this patient is not the same if you receive a transplant or not, or if you receive maintenance or not. And the third study can be in the future even more relevant because it has been shown in this study that in patients with deletion of chromosome 17 plus chromosome 1 amplification, there is a protein that is involved in DNA repair called CHK1, and this protein is highly overexpressed and can be a target for a specific treatment in the near future. The second topic, moving from genetic, that I want to discuss with you is the evaluation of the response to therapy. All of you know that response to therapy is key regarding the prognosis. If you respond well, the prognosis is much better as compared if you have just a suboptimal response. Until now, we were using for response, the better criteria was a complete response, the disappearance of the N component, and to have less than 5% residual plasma cell in the bone marrow. But these residual plasma cell in the bone marrow were analyzed by morphology. And morphology does not allow you to separate between the normal plasma cell and the malignant plasma cell. Is this for the reason why we have implemented new techniques to discriminate between the normal and the malignant plasma cell, and this can be genomic techniques, next-generation sequencing, or antigenic techniques, immunophenotyping, next-generation flow. And these techniques allows you to identify one residual myeloma cell, tumor cell, among 1 million of total plasma cell. And it has been demonstrated that the true value of achieving a complete response is not the disappearance of the N component or to have 3% plasma cell, is to have a good level of minimal residual disease, a low level of minimal residual disease. And does matter, the level of minimal residual disease clearly matter. This is like an iceberg. The lower the level, the longer the survival. And this has been shown by both techniques, next generation sequencing and next generation flow. As you can see, the highest survival is achieved for those patients that show less than 1 cell in a million cells in the bone marrow. Does this type of analysis have impact in all patients? The answer is clearly yes, but particularly in the high-risk patients, in those patients that have a stage 3 or high-risk cytogenetics. And the reason is because in these patients, either you get rid of the tumor cells or you will have always a problem of early relapse. then to have these techniques to measure the efficacy of your treatment is most important. And at this meeting, several abstracts has been presented that already analyze more and more patients and demonstrate the efficacy of these techniques and the prognostic impact. In one of them, it was shown that achieving MRD negativity is associated with significantly lower incidence of progression. The UK study, a very large study, demonstrates how one of the treatment adding 4 drugs instead of 3 drugs is associated with much higher incidence of residual disease. Similar, the transplant is also helping you to eradicate the disease, and how the proportion of MRD negative patients decrease with the prolonged treatment. Then, if these techniques are so relevant, should we use to drive therapy? This was a big debate this morning about this, and we know that there are some pitfalls. An MRD negative is not by sure that you don't have disease, because this will depend very much on the area in which the analysis has been done. As you know, the infiltration in bone marrow, the disease has a patchy infiltration. And it could be that if you have the bone marrow analysis in an area that is free of tumor cells, but they are in another area, then the result can be negative and can be a false negative. But if it's positive, and the enemy is when you have a positive residual cells, this is by sure residual tumor cells. And then we are now trying to implement this to try to monitor the treatment efficacy and in order to make decisions for the patients. But we have learned that in addition to this potential pitfall of having an MRD negative not being a true MRD negative, we have learned that it's necessary to have confirmatory data for MRD. And this confirmatory means, yes, to repeat the bone marrow in 3-6 months, just to be sure that it's really negative. It's what we call now sustained MRD negativity. And here I'm showing that through maintenance, in this is a French study, this is a Spanish study, while the patients were in maintenance therapy, they remain MRD negative, but as soon as they stop the maintenance, they start to relapse. Then there is another piece of information with this slide that illustrates you how important is to monitor and how important can be this to make good decisions about for how long to maintain maintenance. Maintenance is part of your treatment, it's very important, and again, in this meeting, there are several papers that have shown the efficacy of maintenance therapy. And I want to just to highlight that until now we were using for maintenance therapy mainly lenalidomide that has improved for more than 2 years the progression-free survival and the overall survival. But in the high-risk patients, there is emerging data that having a proteasome inhibitor is important in the high-risk patients. And there is one study from UK, another study from the German group, in which they have shown that the use in high risk of proteasome inhibitors may be equivalent. But if it's equivalent, the proteasome inhibitor to the IMiDs, why not to put together? And this, I think, is going to be the future in maintenance for the high-risk patients. And this is my last slide. This is a summary slide. And these are the take-home messages. First of all, myeloma is very heterogeneous. None of you that have multiple myeloma are identical to the other one. And for this reason, we need to individualize the monitoring of the disease and the treatment decisions in the future. We have now genetic information that predict the risk of progression. We know that some of these abnormalities should decide in the future for treatment decisions, and the high-risk patients should not be treated in the same way as the standard risk. Standard risk should receive the optimized treatment, the most intensive possible treatment, but the high risk, we need experimental therapies for them. To achieve MRD negativity is key, and in fact, is the only way for a high-risk patient, yes, to have a prolonged survival. And for this reason, it's now an endpoint in many of the clinical trials. And as I have shown, maintenance therapy is important in myeloma, similar to other leukemias. In chronic myeloid leukemia, we have learned that to maintain Gleevec, that is the drug for this leukemia, for a long time is important. But now we also have learned that at some time point you can stop and you can have a free interval without the drug. And if the patient has a molecular relapse, you introduce again the drug. Then I think we are moving in this direction for the myeloma because our goal is to cure or to make at least really a very chronic disease. And thank you very much for your attention.
Hello, good afternoon, everyone. And well, first, thanks to the Myeloma Research Foundation for inviting me to be here with you, because, well, we don't have any doubt that we discuss and we study a lot of multiple myeloma, but the key players of the disease are you, and you are here, and, well, it's really a pleasure for me. Following a bit after the introduction that Jesus and Miguel did, and Jesus is my mentor, I started in the myeloma when the new agents appeared. And in fact, I remember very well the first patients we treated in Spain, in Salamanca, with bortezomib. And as this occurred, well, in the last century. And right now, I think that the options of therapy are really, really very, very big. And well, Irene will discuss later on about the management of symptomatic myeloma patients, newly diagnosed myeloma patients, and from MMRF started her talk saying that our dream and our objective is to cure patients. And you are going to see how the highest chance to be cured may be occurred in the upfront setting. And Irene will show you very nice data showing how newly diagnosed myeloma patients are achieving right now high rates of complete response, high rates of minimal residual disease, and how this is going to clearly impact not only in the progression-free survival, but also in the overall survival. But it's true, and this is the real life, that not all patients are going to be cured. And this is the reason why my talk is going to be based on the management of relapsed and refractory myeloma patients. And I'll go back to the first slide. And when we have in front of us a relapsed and refractory myeloma patient, as a physician, we have to consider what do we know about the patient's myeloma. And I always recommend to my fellows, to the young hematologists, that it's important to take a paper and to write what prior therapy the patient received, what was the response the patient achieved, what was the duration of the response, how was the biology of the disease, which information do we have about the biology of the disease at the moment of diagnosis, but also it's important to repeat some biological studies, the FISH, the cytogenetic information at relapse. And with all this information, I think that we have also to consider the patient, as I previously said, the key player of the disease. And we have to consider features coming like the age, or the prior medical history, the comorbidities, hypertension, diabetes. How was the residual toxicity the patients present coming from the prior lines of therapy, like a residual peripheral neuropathy or any thrombotic event? How is the bone marrow reserve? Some patients arrive at the moment of relapse with a poor bone marrow reserve. All this information is key when we have the myeloma patient at relapse in front of us. But finally, and this is really very important, we have to consider the patient preferences and values, because we can't forget that you are the key players in this game, and we have to hear you, and we have to discuss you the different options of therapy, and our thoughts with your preferences will make the right choice. And I am saying this because this is a busy slide, and I am not going to enter into detail, but it's just to explain, and this is just to say how there are multiple options of therapy that we can potentially use at the moment of relapse. And today, in 2019, you have to know that the first line of therapy, and Irene will discuss about that later on, is going to influence the rescue therapy. And I previously said it's very important to evaluate what was the prior line of therapy and what was the response achieved and the duration of the response and so on. And at early relapse in this slide, you can see here like we can use proteasome inhibitor-based combination or IMiD-based combination, and we can select lenalidomide dexamethasone-based combination or pomalidomide dex-based combination. And for later relapses, it is also possible to use some combinations based on pomalidomide, although it's true that we are going to see new agents with different mechanism of action. And well, finally, and Paul will discuss about this in his presentation, new immunotherapy agents that personally I consider that maybe they are going clearly game changers, and he will show you the most exciting results. But now we have just finished our international meeting focused only in myeloma that we celebrate every other year. And I would like to share with you first the updates on existing agents coming from this Congress. And there are, and we have selected here three relevant drugs. Ilotuzumab, implicity, is a monoclonal antibody targeting a specific antigen that is present on the surface of the plasma cells and also in an immune cells called NK cells. Maybe you know, all of you, ilotuzumab in combination with lenalidomide and dexamethasone. And it is an approved combination because a phase 3 randomized trial was conducted comparing ilotuzumab, lenalidomide, and dexamethasone with lenalidomide and dexamethasone alone, and the results were presented some years ago. And the addition of ilotuzumab resulted into a significant benefit in terms of progression-free survival. This means that when patients received the 3-drug-based combination, the duration of the response was much more longer. But it's important to update all these trials, and this trial maybe started more than 6, 7, 8 years ago, and now in this Congress we had the opportunity to see how this combination resulted not only in a benefit in terms of progression-free survival, but in overall survival. When patients received that relapse, ilotuzumab plus lenalidomide and dexamethasone, they are going to live longer. What is relevant, because this is the final objective we have to try to achieve with all combinations. So this is an important data. If we focus on carfilzomib, you know that is the second generation proteasome inhibitor, and Irene maybe will discuss because there are some trials trying to move the second generation to the first line of therapy. But during this Congress, we had the opportunity to see how to manage carfilzomib in the elderly population, and especially in a modified dose. While here in the US, the conventional dose is 27 milligrams per square meter twice per week. It was the standard of care. But we realized that this was not convenient at all for the patients because they have to come to the hospital twice per week. We decided to move to the weekly administration, and we increased the dose up to 70 milligrams per square meter. In this Congress, we had the opportunity to see how 70 milligrams per square meter, the higher dose of carfilzomib, given just once a week, was more effective than the twice-weekly administration. But when we focus on the elderly population, there is not any major concern in terms of efficacy and in terms of safety profile. And this means that the elderly patients can safely receive the carfilzomib once per week at a dose of 70 milligrams per square meter, and as 70 is a high dose, your physician can potentially modulate the dose and even to go down to 56 if the medical condition allows or don't allow to receive the higher dose, 70 milligrams per square meter. The third drug is pomalidomide, and concerning pomalidomide, here in this Congress, we had the opportunity to see a couple of new updates coming from different trials. The first one is relevant, is pomalidomide in combination with bortezomib and dexamethasone. This is a new combination that we can use in relapsed and refractory myeloma patients just since the moment of first relapse. And this trial was conducted in patients that all of them had been previously treated with lenalidomide. And some patients, approximately 60%, had been previously exposed to bortezomib. And patients at relapse were going to receive again pomalidomide, bortezomib, and dexamethasone. The prior exposition to bortezomib could potentially influence in the efficacy of this combination, and Paul Richardson, Thanos Dimopoulos presented this data, and the answer is no. If you have received prior therapy with lenalidomide and even bortezomib, you can receive as rescue therapy pomalidomide, bortezomib, and dexamethasone because you are going to respond again. And this combination resulted very effective, and in fact, PVD, pomalidomide, bortezomib, and dexamethasone showed in this update 53% reduction in risk of disease progression or death compared with bortezomib and dexamethasone alone. And as I previously said, we are moving towards the second generation proteasome inhibitors, carfilzomib. And Professor Jakubowiak from Chicago presented a study in which he decided to combine patients treated with, again, pomalidomide, but in combination with carfilzomib, the second generation proteasome inhibitor. And this combination was also highly effective. You have to know that this combination was evaluated in late advanced stage of the disease, so the number of prior lines of therapy the patients had received in this study was 4, but this combination is effective, and it is possible to use as rescue therapy, maybe not today at first relapse, but second relapse, third relapse, even although you had been previously exposed again to lenalidomide or to bortezomib. The latest drug for relapsed and refractory myeloma patients is selinexor, and selinexor is a new agent, but it's important you to know that the mechanism of action is different, because what this drug does is to accumulate, to have a mechanism of action that the final goal is to accumulate within the nucleus of the cell many proteins that are toxic for the cell, and the cell finally die. This mechanism of action is very interesting. And this drug, in combination with dexamethasone, has just been recently approved by FDA here in the US. Why? Because this new agent was first evaluated in patients at late advanced stage of the disease, what we call triple class refractory. This means that patients had been previously exposed and refractory to proteasome inhibitors, immunomodulatory drugs, but also monoclonal antibodies. Monoclonal antibodies targeting CD38. In this population, when they are refractory to everything, this population represented an unmet medical need. And selinexor plus dexamethasone showed to be effective in this population. And approximately 1 out of 4 patients responded, and the authorities here in the US considered that these results were exciting, and this is a new agent the patients here in the US can receive. But as the clinical research is moving forward, selinexor is being combined with the different backbones, with the different standard of cares. And there is a phase 3 randomized trial in which selinexor is being combined with bortezomib, and we don't have the results yet, but during this Congress we had the opportunity to see preliminary results when selinexor was combined with lenalidomide. And in the slide you can see here, like, the overall response rate is not anymore 25%, but over 90% when we combine a new agent like selinexor with a standard of care, in this case lenalidomide, and we are going to see more trials and more combinations based on selinexor plus the monoclonal antibodies, proteasome inhibitors, and so on. New drugs and new myeloma targets from International Myeloma Workshop 2019. Venetoclax, melflufen, and AMG 176. I would start by venetoclax, and venetoclax targets BCL2, that is an anti-apoptotic protein. BCL2 as well as MCL that you can see in the right part of the slides are proteins that are going to prevent the plasma cell die. When we inhibit these anti-apoptotic proteins, the final result is that we are going to stimulate, or we are going to enhance the kill of the myeloma cells. Venetoclax was evaluated in combination with bortezomib and dexamethasone in a phase 3 randomized trial, the Bellini study. And this trial showed how when we combine venetoclax with bortezomib and dexamethasone, the patients responded more, and the complete response rate were higher, as well as the minimal residual disease negative rate, and also the progression-free survival. However, it's true that there was observed an excess of deaths in patients receiving venetoclax, bortezomib, and dexamethasone. The explanation for this is not clear, and safety is not the main issue, and we are investigating why this occurred, and what we know right now is that BCL2 inhibitor venetoclax targets patients with age-specific translocation 11-14. And in this population, the benefit was maintained in terms of progression-free survival, and the absence of benefit in overall survival was not observed. This is the reason why my message concerning venetoclax is that the upcoming trials are going to be conducted in patients with 11-14 translocation. This would represent a targeted therapy, and we will see the final results, but maybe we can use venetoclax in this specific population. AMG 176 is a MCL1 inhibitor that is also, MCL1 is coming or is becoming from the same family from BCL2, and during this Congress, a phase 1 clinical trial was reported, the first in human trial with this drug. So the results are very preliminary, and just to say that the drug is safe, and it resulted to be effective in patients after a lot of number of prior lines of therapy, and we have to wait. And in the middle, you can see also a new agent with a different mechanism of action. What is important for us as physicians when we have the patients at relapse in front of us, to try to find drugs with a different mechanism of action. And this is melflufen. Melflufen is an alkylator agent that it is going to target aminopeptidases, and aminopeptidases are overexpressed in the plasma cells. So this means that it seems also a targeted therapy focusing on plasma cells, and melflufen is going to result in the killing of the plasma cells. Melflufen has been evaluated in over 100 relapsed and refractory myeloma patients that they had been received proteasome inhibitor IMiDs, and in fact, patients were refractory to pomalidomide and or daratumumab. And in this situation, melflufen works very well. But during this Congress, Paul Richardson focused on the presentation based on the results with this drug melflufen in a specific situation that, believe me, that is really challenging for physicians, extramedullary disease. When a myeloma progresses with soft tissues around the bones or outside the bones, the situation is really challenging. And this can occur in up to 30–40% of the patients at the moment of relapse. In this presentation that Paul did today, this morning, in the late-breaking abstract session, melflufen has demonstrated to be effective in this population with extramedullary disease. This is relevant for us as physicians. This is relevant for you as patients, and in the future we will generate combinations of melflufen, again with the different standard of care, including the monoclonal antibodies, and definitely melflufen will be incorporated to the treatment armamentarium, and it is planned to submit the submission for approval by FDA at the end of this year. This is my last slide, just to summarize that new combinations of available drugs can help patients previously treated with Velcade and Revlimid. Pomalyst-based combinations are being moved to first relapse. Venetoclax may still be used in patients with 11-14 translocation. Novel cellular pathways are being targeted for myeloma treatment, including patients with extramedullary disease. And my last sentence is that clinical trials are the optimal way to answer any question. Don't hesitate to be included in clinical trials if your physicians offer you to be included. This is the only way in which we can improve, and we can improve the knowledge of the biology of the disease and how the treatment can evolve, and the final objective is to cure patients with multiple myeloma, so please don't hesitate to collaborate with us. And I stop here. Thank you very much.
Okay, thank you so much. Just a couple quick notes. We meant to say at the beginning, we switched the agenda so that our Spanish friends could catch their plane. So we'll do a short, focused Q&A right now. I saw a lot of the questions coming in were more around immunotherapy, so during the second session with Paul and Irene, we can do those then. But I thought there were a few really interesting questions that were very suitable for a discussion, particularly around MRD. So one question that was asked, and I asked different members of the panel to chime in, how reliable is MRD testing? How can we be sure that we're not missing myeloma cells that just weren't in the sample?
Is this working? Oh, it's working.
Maybe this one.
It's working, man. Okay. As I emphasize, for me, what is very important is the concept of MRD positivity. Because with MRD negativity, you may have a false result due to the quality of the sample. But if the cells are there, if you identify the cells, then the enemy is there, and you have the opportunity to fight and to try to eradicate these residual cells. There is one exception to this situation when you have minimal residual disease. In the rest of the cases, always minimal residual disease predict for shorter survival, but there is an exception. Are those patients that have a very strong immune system that is able to control the residual cells because they have returned into an MGAS profile. And we are also working just to identify this situation.
And then go on, Carl.
Can we add a little bit more to that? I just think this it's a very, very important point. It came up very strongly in the debate. I'm so sorry. Can you hear us now, guys?
No, here.
Oh.
No, the microphone.
It's not this one. Sorry, man. I apologize. Anyway, so basically, the challenge in the debate today was really that I think MRD clearly as a research tool and as a device that allows us, or a modality that allows us to get rapid regulatory approval for new drugs is clearly established. The challenge is in clinical practice, how does it change fundamentally what we do? And I think Jesus did a beautiful presentation this morning, but was very firmly challenged by a very seasoned clinician, Sundar Jagannath, who leads the center at Mount Sinai, to say, well, steady on, because at the end of the day, we don't know exactly how to change therapy at the moment in every case on the basis of MRD. And I think we have to be careful about this, because I absolutely agree with Jesus. There is this subgroup of patients in whom their immune system resets, and they seem to tolerate a minimal burden of disease with remarkable equilibrium. Personally, I think the MGUS reset is perhaps a misnomer, because I think it's not really an insignificant reset, but that works both ways. It doesn't work just in the sense of it being a relatively small number that's indolent. I think it also is something we need to much better understand, because in my practice, I have a large number of these patients who are very stable, 10 years, 12 years, on IMiD-based therapy, and they do very well. And their immune paresis is minimal, as is to the point. But I think we really need to understand this better, because I am also struck that as we strive for MRD negativity in some patients, we see a very rapid relapse that then follows, despite the fact that they're MRD negative. So sometimes there's a little bit of caution that needs to be exercised in understanding exactly what this means. But broadly, I absolutely agree with Jesus that certainly in the context of clinical trials and our ability to gauge what intensity of therapy may be needed, MRD constitutes an important tool.
And a good, I think, next question is, and there may be some differences of opinion here, if a patient is MRD negative and flips over to MRD positively, do you treat right away?
It's a great question. Jesus.
Depends very much. If the patient is high risk, I will act. Yes. If the patient is a standard risk, no, I will be prudent.
Yeah, I think we're all giving you the same message. We still need more answers than what we have now. We know that the depth of response is very important. MRD is basically measuring for us even deeper response than what we call complete remission. But we know that there is a second part of that. It's not just the cancer cells that we're trying to kill, but we're also trying to make your immune system better. And if we can see that immune system improving, even if people have still some residual cells, what Paul and Jesus and everyone was calling MGUS-like, which is what we're trying to define as small number of cancer cells are still left behind. We can still see them, but your immune system is able to control it and put that balance back again. So we still don't have all the answers, but the best thing is that when you're in a remission, whatever the remission is, for a very long duration, that's what matters, is that sustained time in a remission.
And I would like just to add that together with the minimal residual disease evaluation into the bone marrow, myeloma usually affects also outside of the bone marrow. And now we have new imaging assessments like PET-CT that are going to be incorporated into the measurement of minimal residual disease. And the final objective would be patients to achieve MRD negativity inside of the bone marrow, but also outside using, for example, PET-CT. And we have some experience coming from some clinical trials, and when patients achieve the MRD negativity inside and outside the bone marrow, the outcome is much more better.
Okay. And a couple of questions around transplant. One, and the question, the person uses the term CR, but whether it's MRD negativity or not, if a CR is achieved without transplant, would you recommend transplant at relapse?
Yeah. No, it's a great question, and it was one of the major topics of debate this morning, which actually was won very firmly by Dr. Ola Landgren from Memorial Sloan Kettering, where 30% of the audience were in a sort of cautious, or rather advocating for transplant. 70% were saying it could reasonably be kept in reserve, which was, I think, quite a sea change. I think the important message with the transplant construct is that if the therapy is destined to deliver absolute benefit with no harm, that's essential. Mm-hmm. That's the challenge, because if you drive for MRD negativity, but you do so at a high price, not only in terms of toxicity, quality of life, but long-term side effects. And most importantly, there was some extraordinary data presenting on whole genome sequencing showing patterns of relapse after melphalan exposure are different genomically than they are if you don't get melphalan. So I think we have to be very careful, because Mary V touched on an extraordinarily important point. The venetoclax studies told us just because you have a high quality response, just because you have PFS advantage, guess what? That trial was stopped because there was no overall survival advantage. In fact, went in the opposite direction. And I think in that sense is an important lesson that we have to be very careful how we intervene and look at the long-term consequences of the same.
Yeah, so we were discussing this morning about the role of transplant. And well, in Europe, there is not so much debate about the transplant because we usually offer transplant to every newly diagnosed myeloma patient that we consider the patient is transplant eligible. It's true that we have 2 phase 3 randomized studies, 2 large phase 3 randomized trials in which transplant has demonstrated to be better than non-transplant in terms of progression-free survival, so in duration of the response, I would say. But none of these trials have shown so far benefit in overall survival. From my point of view, this is relevant because, again, you are the key players, and maybe you can ask us, and in absence of benefit in overall survival, your value and your opinion is going to be important. And I think that in the future we are going to conduct more and more trials in order to try to describe or to identify if we can potentially refer the transplant at the moment of relapse in some patients. And I think that together with the genomic studies, I think that the risk factors, patients at standard risk, ISS1, with no cytogenetic abnormalities, if they achieve minimal residual disease negative after an excellent induction base, maybe in 3 drug-based combination plus monoclonal antibody, maybe these patients can potentially not receive the transplant in the upfront setting. And this is, I think that the work is in progress, and well, Francesca Guy is evaluating this in a trial with carfilzomib, lenalidomide, and dexamethasone, and I think that we need more trials in order to really be confident when we have the patient in front of us and to say this, well, this patient don't need a transplant now in the first line.
Yeah. I would like also just to say that high-dose melphalan, because we call transplant, it's not a transplant. It's high-dose melphalan with stem cell support, which is clearly different. High-dose melphalan has demonstrated to be highly efficient in multiple myeloma and is a strategy that particularly in countries that they have no access to all the drugs, it has been able to demonstrate long survival. Look, at the last ASH meeting, it was presented an Italian and Spanish study in which patients had long-term follow-up. There was more than, both trials started in 2005, 15 years follow-up. And the progression-free survival with VTD plus autologous transplant plus thalidomide maintenance, old-fashioned, the progression-free survival at 10 years, this means you have not progressed during 10 years, is 30%. And is potentially this patient may have been cured. Then the name of the cure is coming and coming more frequently, and the transplant has played a role in this. What I say this is because if in our center, if your doctor offers a transplant, be sure that this is an efficient, very efficient treatment that reduces the tumor load significantly, but we are trying to investigate other alternatives since we have realized that for some of you it's toxic at least for a period of time, and this is how we are working on other alternatives, potentially.
So I'll do one final question, which is a fun one based on the composition of the panel. Tandem transplant. Is there a role? Yes, no, and would love to hear the discussion.
I would say no, unless very few exceptions.
Yes. I would say in the United States, and as Jesus makes an extraordinarily good point, we're so lucky in the US that we have access to good salvage options that are relatively non-toxic and highly efficacious. We very rarely, we do not do tandem transplantation outside of a clinical trial, and we haven't had a clinical trial in this space now for almost 10 years. And so I would have to say we do not advocate for that except under exceptional circumstances.
Did you ask by tandem? Tandem
yeah.
transplant. Well, it's a controversial topic. There is a big analysis, because this is not a trial, a big analysis conducted by Michele Cavo from Italy, and he decided to put together approximately 1,000 newly diagnosed myeloma patients included in different trials coming from Spain, France, Italy, and Germany. All patients had been previously treated with bortezomib-based combination, and approximately half of them have received a single transplant and half of them tandem transplant. When he evaluated the outcome of these patients, tandem transplant was better than single transplant in terms of progression-free survival and also in terms of overall survival. And when he evaluated in detail which patients could potentially benefit most from tandem transplant, he realized that patients having high-risk cytogenetic abnormalities benefited the most from tandem transplant. This is the reason why in my practice, and I think that this is more or less general in our country and even in Europe, we are offering tandem transplant to patients with high-risk cytogenetic abnormalities.
And if, I'm so sorry, just for our audience, just so we know that this randomized trial that we did in the US, using US-based practice, did not show a benefit to the tandem transplant, just to be very clear, because and
Exactly.
even in the high-risk patients, because what I think is so true is one size does not fit all. And in the European practice, you're seeing from Jesus and Marie V that they have certain imperatives. They don't have choices as we do in quite the same way. And so as a result of that, practice patterns may be
Absolutely
different.
right.
Yeah.
As Marie V has mentioned, in Europe, we propose tandem transplant for the high-risk patient, but with an exception.
No.
If you have a first transplant and the response to the first transplant is suboptimal, even if you are at high risk, in my clinical practice, I will not offer outside of the clinical trial the second transplant. By contrast, if you're at high risk and the first transplant has worked very well and has already going to minimal residual disease negative, I will be confident to offer the second transplant. Then once again, to individualize this offering to the patients, I think is extremely important. Let me also add another possibility. The tandem auto-allo transplant. We don't offer allo transplant to our patients, but I should recognize that in this meeting, it has been presented a very interesting study with tandem auto-allo in high-risk patients with so far very positive results, very good results, and a mortality rate that was impressively low, was less than 6%, which is clearly, in the long term, clearly different from what is expected. Once again, what I say this is because sometimes you may talk with a doctor, your doctor say you have 3 cytogenetic abnormalities plus blah, 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 and you have an early relapse, and you are now negative for, and I will offer these possibilities. This could be reasonable. I will not be nothing against.
I think it raises such an interesting point, though, because I think what you're hearing, because I had the privilege of working for an amazing man called Tim McElwain at the Royal Marsden in the late 80s, early 90s, and his rationale was high-dose melphalan and immune therapy with gamma interferon after transplant. It was a brilliant rationale 30 years ago, because what he was trying to do was cytoreduce and then introduce immune manipulation. And what you're hearing, I think, from Jesus is exactly that, using the tools that are available in that setting. And what we hope to do is do exactly the same thing, cytoreduce, control, restore immunity, but hopefully not with the potential risk of the surround sound side effects that come from a less targeted approach. And I think just to help you make sense of that, because we certainly don't routinely do allogeneic transplant for our patients in the US, as is at our center anyway. From the Dana-Farber perspective, the use of allo transplant is very limited for myeloma.
Okay. We don't use as well, but I was really favorably surprised with the results.
Well, thank you. We will let Dr. San Miguel Mateos get to their plane. There'll be another session after Dr. Gabriel and Dr. Richardson speak. So Dr. Gabriel will now talk to us about smoldering and newly diagnosed myeloma treatment strategies.
Beautiful. Great. Thanks, Jesus.
All right, so we'll go back to the upfront setting and thank again our European colleagues and hopefully safe travels back home. So what I'll try to take you through a little bit is some of the updates that we had for MGUS and smoldering myeloma and some of the debates there, whether we should treat or not, and then updates on upfront myeloma and some of the new studies that we have there. So just to remind everyone that myeloma is truly a spectrum and that almost every patient who has been diagnosed today with myeloma must have had MGUS and smoldering in their lifetime before. But because we don't screen for it and we don't look for it, we never find it until you have fractures or anemia or kidney failure. And we know that basically the asymptomatic stages are two stages, MGUS and smoldering myeloma. MGUS is the very early stage when you have less than 10% plasma cells, when you have a very small amount of protein in your blood. Smoldering myeloma is a little bit higher when you have more than 10% plasma cells. And the risk of progression in smoldering myeloma is 10% per year, but we know that within that group of people, some will progress very fast to active myeloma and have the end organ damage, and some will never progress in their lifetime. And what we're trying to know is define who will truly progress and try to treat you early so that you don't end up having fractures and anemia and kidney failure. We want to stop that from happening. And we want to also make sure we don't overtreat someone else who should not be treated for myeloma. So what are we doing now? We're all working together, multiple centers, trying to understand truly who will progress in their lifetime. And we know that there are clinical markers that we use. So when you come and see us, we say, well, if you have 10% plasma cells, if you have a light chain ratio that's elevated, if you have an M spike that's elevated, then you're at a certain risk of progression from smoldering or from MGUS to myeloma. But we know that these reflect something called the tumor burden, meaning how much disease you have in your body. What we're trying to do is we're trying to say what's the biology of your disease. If your cell is growing very fast, that's very different than someone else, the same numbers, but their cells are not growing that fast. And those cells will keep on staying like this for a very long time. So we try to look at something called cytogenetics and FISH, and that's why we look at all those numbers. And we also look at something called next generation sequencing, going a little bit deeper at the single molecule level to try and see if you have one mutation or the other, or one change in your chromosome that's much smaller than when we look at it by FISH and cytogenetics. And I think that's important to look at, but also to look at your own immune system. And we're doing now work on looking at your bone marrow as a whole, trying to understand that if you have certain cancer cells, but your immune system is still very strong, maybe you will not progress to myeloma very fast, while if you have the same type of cancer cells, but your immune system is suppressed, and we can measure that now, then you may be one of those people who will benefit from early treatment because we know that your myeloma will progress rapidly. So these are all things that we're looking at, and one of the studies that we showed already from looking at the next generation sequencing of the cancer cells is that now we have certain markers that can predict for us progression, like MYC alterations or amplifications, like MAP kinase pathway or DNA repair. You don't have to know all those things, just knowing that we're working very hard to add to the FISH and cytogenetics new molecules, new markers that can predict progression for us. The other thing, and we would love your collaboration and your effort with this, is we have started for the first time the screening study for multiple myeloma. For the first time in the US, we're saying, why are people waiting until they're diagnosed incidentally, until you have anemia or fractures? When you go to your primary care doctor, you get screening for everything else for cancer, because cancer screening hopefully means early interception and early prevention. You go to get a screening for mammography, for colonoscopy, even PSA, that's controversial, people screen for prostate cancer. Yet we have a blood test that's very sensitive and very specific, and we're not using it to test for multiple myeloma. So now we started the first screening study for myeloma, and this is available for everyone. If you have brothers, sister, family members, first-degree family members
who are at risk because you have myeloma or MGUS or smoldering or Waldenstrom, please encourage them to go to promisestudy.org, and we send them a kit at home and they can see if they have MGUS or not. And this is not to make you anxious or to worry you that indeed you have that, but it's potentially to prevent it. If I know that I have MGUS now, I'd better know that and potentially go to my doctor and watch carefully or see if I'm enrolling in a clinical trial, if I'm going to be at risk, rather than not knowing and getting diagnosed with myeloma a few years from now because I did not look for it. So we're really encouraging everyone to try and go to promisestudy.org and try to enroll on it. We're looking for 50,000 individuals, and we're collaborating very closely with MMRF and MMRC on this effort. Then I'll take you through the upfront treatments. And remember that right now when you're newly diagnosed, when you have anemia or kidney failure or lytic lesions, when your doctor tells you you have myeloma, we need to start therapy, there are so many options of treatment. And in fact, compared to the old days, compared to 10, 15 years ago when we only had 2 drugs or 3 drugs, now we have so many amazing drugs. So I'll take you through sort of themes of what we saw this year. We saw a couple of interesting themes. One is 4-drug regimen is coming these days for treatment of upfront myeloma. So remember, right now we use 3-drug regimens, RVD, lenalidomide, bortezomib, and dexamethasone, or KRD in some other centers, carfilzomib, lenalidomide, and dexamethasone. Now we're starting to say, should we use 4 drugs to even improve more on what we do, so that the response rate is not just 100% response rate, but it's a deep response and it's a very long remission. And that comes into play whether we should transplant patients or not, as the remission is getting deeper and deeper with all those 4-drug regimens. And the biggest highlight here was Darzalex is coming up front in the treatment of newly diagnosed young patients, whether they're eligible for transplant or not. And this has been shown in multiple studies. One of them was presented today in the morning called the Griffin study, where we're using daratumumab with RVD, all of them 4 drugs together, showing us a very deep remission in our patients with upfront myeloma. So I think that's one message to remember, that we're starting to look at 4-drug regimen in many of you who are going to go for therapy. The second message was, we're trying to improve on our toxicity. We're trying to get better in giving you the same 4 drugs, but with less toxicity, which means that if we're giving you carfilzomib, we noticed that giving it to you 2 days in a row, which is the twice a week, is as good as giving it to you once a week, but with a higher dose. And in fact, now most of us in the practice, including us at Dana-Farber, we're switching to that once a week carfilzomib rather than giving it to you twice a week. We found that the toxicity is very similar, although it's a higher dose, but in general, once a week is better, and it gives us that high response rate that we're hoping to look for. So again, ask your doctor, if you're using the twice a week carfilzomib, should I consider the once a week carfilzomib? The other new thing that will come hopefully soon, once approved, is we're switching from IV daratumumab, where you sit in the infusion room for hours and hours, into subcutaneous daratumumab. And I think that's amazing because it will be much faster, just a few minutes of subcutaneous injection, and it will be much easier, less toxicity, very convenient for everyone. So we're really looking forward for the subcutaneous daratumumab. And then finally, real-world data. We are looking for you telling us what is tolerated and what's not tolerated and how to improve on that. So here's just that data of the 2 trials that were presented with the 4-drug regimen, and I've highlighted already the daratumumab, Revlimid, Velcade, dexamethasone, and how this will truly change the way we treat our patients. We saw this data today, and I can tell you now we're starting to see our patients in clinic this week, next week, and we will tell them there is the data of the 4-drug regimen giving us high benefit compared to 3-drug regimen. And this is the data for the once a week carfilzomib that I was just talking about, giving us this depth of response, same toxicity or improved toxicity, but giving us very high response. The subcutaneous daratumumab showing us that indeed you can use subcutaneous daratumumab and it's very well tolerated and it's not as toxic. And then real world data, large numbers of people telling us maybe we don't tolerate Velcade continuous like this because it causes neuropathy. Maybe we get too much fatigue. Maybe we cannot stay on maintenance for a very long time. These are important messages we're getting back from you so that we can change our message back. We know now that we we have to be careful when we give you long-term Velcade, for example, that can cause neuropathy, fatigue, other problems that you cannot stay on it, and maybe switching to oral ixazomib or switching maintenance therapy to something that's well-tolerated could be easier for our patients. And then the other things that we talked about is in the transplant ineligible patients who are usually the older patients or patients who could not go for transplant, what is going on and what are the updates for them. So here what we're trying to say is we're improving on the therapy. And there were a couple of interesting topics here talking about Revlimid-based therapy, meaning lenalidomide and dexamethasone is the base that we're using right now. Can we improve on it? And again, in people who are not transplant eligible but are fit and are doing well, can we use 3 drug combinations, whether it's RVD or daratumumab with Revdex, and improve response and improve survival but still have less toxicity? And I think that was a very clear message from some of this data. The other one was actually looking at all the big phase 3 trials that we've done using lenalidomide and dexamethasone, whether it's something called the FIRST trial, which was done in France, and other trials looking at daratumumab, Lendex compared to Lendex, trying to put all of those together and showing us that indeed you benefit from using lenalidomide upfront in the older population who is not going to go for transplant. And again, going back to the compliance with bortezomib, which is what I was just saying, that real-world data trying to say that Velcade may not be tolerated for a very long time. Should we switch to the subcutaneous once a week? Should we switch to oral ixazomib? Should we look at easier, well-tolerated ways to treat you for combination therapy in the older population? So I know that this was a very short presentation, but we're hoping to have more time for the questions and answers more than anything else. I think just the take-home messages from the early treatment is we are working on improving our risk stratification for smoldering myeloma. Not every smoldering myeloma is the same way. We know that there is a lot of controversy whether we should treat you or not for smoldering myeloma, but this is, again, with clinical trials, we would improve on it and potentially treat early so that we can prevent fractures and anemia. We're hoping that you will be part of the PROMISE study for the screening, and again, this will be also part of the Cure Cloud, so that it's important to put your data into the Cure Cloud so that we can understand better how to treat you and have precision medicine in the future. Not every patient with myeloma should be treated the same way. And we know now that upfront myeloma therapy is getting to the 4-drug regimen, improving response, adding the antibody early on, like daratumumab, to our treatment so that we can get deeper and deeper remission. And then in the older population, lenalidomide and dexamethasone with a 3rd drug gives us that improved response. And we're looking at improved toxicity, better tolerability. Carfilzomib once a week, subcutaneous daratumumab is coming into our world so that we can improve on the response as well as the toxicity for our patients. So with that, I'll stop and I'll have Paul Richardson come along.
Thank you very much, Irene. That was lovely. And I think the important thing for this next part of the discussion this afternoon is to really thank you for coming on a Sunday afternoon to come and hear about what we've been doing for the last few days. But it's really a privilege for us to share this with you. As you heard very nicely before during the introductions, this was the 17th International Myeloma Workshop. It's historically interesting to note that the first one was held actually by the gentleman I referred to, Dr. McElwain, who was a giant in myeloma in his day. It was held at a pub in west of Oxford. There were 18 people, but there was a fun bit to it, which was they had supper at Blenheim Palace, which is, of course, the ancestral home of Winston Churchill. But that was the first one. And so now in Boston, we had 3,200 folks show up, actually. We had over 600 abstracts, and that's something I'm very proud of, in fact, and it was lovely to have Irene chairing this and Murray V with us as well, because it reflects an important point. We had the largest representation of female faculty in any workshop to date, and I think that's very important, so that's good. And I think that it reflects the extraordinary good fortune we have in myeloma, that it's a remarkable global team. And I want to especially thank Anne Quinn Young and the MMRF team for pulling together an international panel, especially there's no one better than Jesus and Marie-V from the European perspective to show you what happens overseas and give you an idea of a different perspective, which I hope is very helpful. Well, with that in mind, I wanted to introduce you to the immunotherapeutic approaches to myeloma, because I think what I was very struck by at this meeting is, on the one hand, our immunology is really going leaps and bounds. It's getting to that goal that Jesus touched on. You know, you use high-dose melphalan to cytoreduce, you try and reset autologous immunity as best you can, and then you use an allotransplant. That was a very legitimate strategy, I would argue, 10 or 15 years ago. Now, with these newer tools, which in my view are better, they're more selective and less toxic, and certainly have enormous advantages in that regard. We have a real, I think, revolution awaiting us as we re-harness the immune response to take on this disease. And you heard very nicely from Ann Quinn Young earlier that immune therapy or immune oncology is doing extraordinary things in solid tumors. It's doing great things in leukemia and lymphoma, and we're doing the same in myeloma. One challenge in myeloma, though, is, in my view, it's the queen of immunological cancers, and it tries to dodge us in every way. So I don't think it's going to be one single immune therapy versus another. I think it's going to be a whole combination of immune therapies, plus, actually, small molecule inhibitors and other drugs. So we have a sort of, literally, my patients will forgive me for this, the Army, Navy, Air Force, Marines, Special Forces, Coast Guard. I think we're going to need a whole lot, because I think, to be honest, to imagine that it's just going to be one element of that probably is too simplistic. So with that in mind, I just wanted to touch on some principles. Why do multiple myeloma cells still grow and survive if the immune system is ready to attack? It's a great construct. Well, basically, we know that essentially, as I mentioned, myeloma, she's the queen of immune cancers. These cells arise from normal plasma cells and therefore may not look like invaders. This is really very true. They throw up disguises. You know, it's like, I don't know if anyone watches Star Trek, but the bottom line, it's like, you've lost these sort of Klingons throw on these shields so you can't see them. And that's literally what happens. And then myeloma cells then fool the immune system by disguising themselves, as I mentioned, in ways that let them evade the Starship Enterprise. And they can actively resist the immune system, and this is the dangerous bit, that they actually are able to produce substances that shut down cell-mediated immunity. And obviously, we all know this as patients and caregivers. Immune paresis is such a feature of this disease. Normal immunoglobulins are down. Infections are such a common problem. And there's a big reason for it, because the myeloma is, shooting a torpedo right amidships to your immune system. So with that, how do we fix it? And I'm sorry, the good news is that we're blessed with a real armamentarium. We have the ability to directly go after the myeloma through monoclonal antibodies. And what's really exciting is older drugs, as we saw them, for example, good old thalidomide, the mother of the IMiDs. That's great. Sounds like my French bulldog. So anyway, the thing is that we had thalidomide that we thought way back in the day was actually an anti-angiogenic. We thought it shut down blood vessels. Well, it did a little. What it really was the first immune therapy. And so in actuality, myeloma has been an immune system targeting disease from a very long time, 20 years effectively. Anyway, IMiDs, a very, very important thing, just a pill, but true immunomodulators. Now, checkpoint inhibitors got off to a somewhat exciting start. In fact, it was wonderful to have Jesus, because he led the original phase 2s that showed some of these checkpoint molecules, or antibodies, I beg your pardon, were actually really doing their job. The snag is, when they were taken out on the bigger scale, there were some real problems, especially toxicities, that precluded any benefit. So checkpoint inhibitors, however, probably do have a role, and we heard at this meeting about some very elegant constructs and models that support that, and some very preliminary data with new types of approaches to checkpoint inhibition that may be relevant. And just to remind you how that works by taking the brakes off the T cells so that it can actually do their job. Now, in that spirit, how can you boost the myeloma-fighting T cells? Well, CAR-T, and we'll come to that in much more detail. But I think what was really exciting at this meeting was also to hear about a different type of cell, the natural killer cell, something that's very important. And as it sounds, it's pretty key. And in fact, harnessing it and getting it to do its job better can be extremely beneficial. So that was exciting. And then finally, how do you sort of give a memory, if you will, or program your immune system to know how to keep the disease from ever coming back? And that's essentially the platform provided by vaccines. So with that in mind, let's talk a little bit about the updates of the immune therapy. So first and foremost, we had some really nice data on CD38 targeting. This has been a game changer in this disease and has really dramatically improved outcome. We have obviously isatuximab, which is originally a chimeric antibody, in fact, different epitope. That means it binds to a slightly different part of the CD38 cell surface marker than does daratumumab. And very importantly, it may have some different activities in that regard that set it slightly apart. But anyway, the isatuximab story got some very nice traction from some subset analyses at the meeting. What was also very important was the BiTEs, and in this regard there was these are so-called bispecific T-cell engagers. What that means is a bit like it sounds actually. There are basically two ways in which the T-cell engages the tumor cell, and it does so by targeting BCMA as the thing that guides it where to go, and then the BiTE brings in two molecules or two cell-mediated components to really to kill the tumor cell. Now, obviously, the chimeric antigen T receptor therapy I've already touched on. We'll go into a bit more detail in a minute. But the CAR-NK cell therapy was really interesting. This was designed to try and get your natural killer cells to behave better and do their job. And then finally, there was this option which I thought was very interesting. For my patients who are on waiting lists for CAR-T, it is really very challenging. And I think also it's extremely onerous that you have to have the CAR-Ts manufactured whilst you're waiting, and you have to get some bridging therapy before it can be given. That's a challenge. Well, the good news about allogeneic CAR-T therapy is that you can do it and have it right there and ready for you. So there's no waiting, bridging, just straight in. So this is very interesting as a strategy. And then most convenient of all are the oral immunomodulatory therapies, or cell mods as we call them. And there was some very nice data on a thing called CC220, or rather CC220. And that data, I know some of my own patients are on that agent. We also have some data also on CC480, which is a more potent version. Okay, so with that in mind, let's talk about isatuximab. If you combine isatuximab with pomalidomide and dexamethasone, and we've been able to show in initial studies that this was very active and that the disease control conferred by it was really quite good. We presented the ICARIA data. This is very much a real-world study internationally done to generate the first randomized data that would provide regulatory approval for this combination of isatuximab plus pomalidomide and dexamethasone. And what we were able to show is that combining pomalidomide and dexamethasone with isatuximab was dramatically better compared to pomalidomide and dexamethasone alone, and very importantly, this translated into improved disease control. So essentially what this did was to generate the platform for an FDA approval that we hope will come hopefully as soon as the end of this year, and give access to isatuximab to patients. Now, what was also very interesting and presented at this meeting by one of my co-investigators on this trial was the combination of isatuximab PomDex in high-risk patients. And what we were able to show, and this was done by actually a very nice Australian colleague, Simon Harrison, to show you the real international flavor of the trial groups we build, that basically if you had high-risk cytogenetics, pomalidomide and dexamethasone was particularly helpful in that regard, and generated an exciting element of disease control that was obviously much better, and in particular in the 414 patients compared to control. Interestingly, in those with deletion 17, the degree of difference wasn't so marked, but I wonder if that's perhaps a sample size issue. We'll see, but certainly in the 414 group, the difference was big. And then finally, co-investigators actually did a very nice study combining isatuximab with KRD in newly diagnosed myeloma, and basically very early data in this regard. But the important point was that the side effect profile was good, and certainly the response rates early were very encouraging, so more to follow, recognizing that isatuximab has already been combined with RVD by the Spanish group. Enrique Ocio, who's actually the nephew of Jesus, who was just here, and Enrique has shown that that's very effective with excellent tolerability. So very nice platforms for isatuximab going forward. Now, what about these famous BiTEs? Well, the bispecific T-cell engagers are basically summarized in terms of mechanism here. And the BiTE bit is that bit right in the sort of middle, which allows basically the myeloma cell to be engaged by the T-cell in a very directed fashion. and it binds through a so-called adhesion molecule called CD3. And so in this regard, in terms of the targeted antibodies that go after it, we're very excited that we have a number of these, and they're listed here. And I will tell you that some of the most exciting data earlyly has been produced by AMG 420, but AMG 701 is the molecule that's going forward, or the antibody that's going forward with Amgen's support. There's some very exciting early data on the Celgene BiTE, looking very, very promising indeed, and there are a variety of others as well. Now, the B-cell maturation antigen, BCMA, is very exciting because its expression is mostly restricted to myeloma cells, and it's becoming a very important target. One thing to be aware of with it, though, is it's relatively downstream in terms of the ontogeny of plasma cells. What that means is there's an upstream population that may be less enriched for BCMA, that are more stem-like in their behavior. And this is important as we think of combination strategies, because it may be important to go after those just as much as it is important to go after BCMA. And we can perhaps touch on that in the question and answer session. So anyway, the data on 420 from the Amgen sponsored trials has been very exciting. This has been predominantly led by the German group, and we heard originally from Professor Topp at ASCO, and at this meeting we heard from his partner, Dr. Hermann Einzeler, who is really quite superb actually, and he reported basically on the updated analysis showing that the MRD negative goal in this relapsed refractory population was actually being seen, and this is really exciting. Now, what's the advantage of a BiTE over, say, a CAR T? Well, the fact is it's off the shelf. The challenge with 420, though, is it's a continuous infusion. And what is true is that the issue that arises is recurrent or significant infections. And in fact, in this trial, just as a word of caution, in fact, there have been some infectious deaths. So we have to be a little bit careful with all of these approaches, but nonetheless, preliminary data for the response certainly is encouraging. Now, the difference to try and get around this is the 701 platform, whereas if you give this less, you don't have to do it by continuous infusion. Perhaps you're able to get around the problem of the inconvenience of a continuous infusion, and also possibly some of those risks of infection and so on, by intermittently dosing this or giving it for a fixed duration of time every month. Suffice to say, preclinically, this drug is or this antibody platform is very effective when combined, especially with the IMiDs. Now let's talk about CAR T, because CAR T obviously has been very exciting in leukemia and lymphoma, curative in those settings. For myeloma, the early data were published by my colleague, Dr. Nuparajeh, published a beautiful paper earlier this year on the BB2121 platform. What was very impressive about what she showed was that whilst a very high response rate, actually the median duration of disease control was about a year. Those patients achieving MRD negativity and actually what was CR, they did a little better. But nonetheless, the relapse rate was real. So there was a challenge here that perhaps it wasn't controlling things quite as well as we would have hoped. In that spirit, there have been a whole barrage now, or a whole number of platforms developed. The BB21217 platform is designed to be somewhat better than the 2121 by virtue of having a sort of longevity of effect that's very important. And in that regard also, there are a whole array of other platforms being studied. And the goal here with the CAR Ts is basically educate these T cells, reinfuse them, and by doing so, engender a smart, targeted immunological approach to the disease utilizing these CAR Ts. So at the meeting, we heard a number of important things. One, that there were some updates, and these from a variety of investigators, but the first here is actually in a Chinese study, and that basically what you see here is that in this very selected group of patients, remarkable activity, everybody doing superbly well, but realize these are very limited numbers in total. Then there was this allogeneic approach that was particularly exciting as well, which may overcome some of the logistical points that I mentioned earlier, which was important. And then finally, I think very interestingly, this NK cell platform, where we primarily saw preclinical data showing the efficacy of this approach, which makes it very attractive. Now, I want to finish up relatively quickly with a couple of slides, but this is a very important antibody-drug conjugate for you to be aware of. What is this? Well, this is GSK 916. It's called Belamaf for short. And what this does is deliver auristatin, which is a chemotherapeutic actually, to the BCMA target with an antibody. So it delivers its payload right in. So to sort of use a military analogy, it's like putting a targeted missile into the enemy and then blowing it up once you're inside. What's interesting about that, especially though, is that when that happens, the tumor cell dies, but it also starts to produce a lot of antigens on its surface that generate more of an immune system, or immune response. So this basically is an immunotoxin which induces strong anti-myeloma effects. Very, very powerful preclinically. Just want to acknowledge my colleague, Dr. Yuzu Tai, in our laboratory, who did some of this early work. Well, what we know from the so-called DREAM-1 study that was published in Blood Cancer Journal earlier this year, and my colleague Suzanne Trudell, who led this trial, and we participated in this, presented this data at the meeting, was that actually we had a very nice response rate from this antibody infused once every 3 weeks. And very, very importantly, these were patients who were very heavily pretreated, and just with the antibody once every 3 weeks, we got a very nice solid response rate overall, but in particular subpopulations, in particular those with daratumumab refractoriness, in other words, daratumumab had failed them, basically we saw an encouraging signal. This drug was working. Most importantly as well, this was durable. So remember I told you CAR-Ts got to about 12 months on average. Well, fascinatingly, 916 gets you about the same in not a dissimilar population, although obviously one has to be careful with cross-trial comparisons. Just to let you know, the DREAM-2 study has met its primary endpoint, and we don't know I can't share with you, unfortunately, exactly the data around that, but because it's privileged, it won't be made publicly available quite yet. But suffice to say, with DREAM-2, we're very hopeful, that's a study which builds on DREAM-1, that there may actually be an FDA approval for belamaf or GSK 916, perhaps as early as the end of the year or perhaps next year. We'll see. In any event, beyond the antibody-drug conjugate, the other bit of data just to be aware of is the exciting new agent, IBRA for short, or iberdomide. I can't even pronounce it, isn't that awful? No, I'm going to stick with CC220. Anyway, so IBRA for short, or CC220. Dr. Sagalonio, who's one of our great partners, he leads the program down at Emory. He presented really nice data showing the remarkable efficacy of this approach in relapsed refractory disease. Now remember, 220 is just a pill, and it's a so-called cell mod, so it's a super IMiD if you like, just a pill once a day, 3 weeks on, 1 week off. And the data were quite remarkable, about a 30% response rate, even in patients in whom daratumumab-based therapy had failed them. How does this work? Well, you see here's actually an example of lenalidomide engaging the so-called cereblon complex, and this bigger molecule engages it more broadly and is much more potent, and in our preclinical systems we demonstrate that. CC480, it sounds a bit like one of these Airbuses, doesn't it? 220 and 480. But anyway, suffice to say, this one's a real big one. By comparison, the 480 is huge. The bottom line is this is a really potent drug, and we're currently studying it. And I think one of the most interesting things to me is just a pill taken every day can be so powerful in terms of its effect on the immune system. So very exciting. So this is my summary. Monoclonal antibodies continue to be an extremely important addition to our armamentarium and consistently improving outcome. I think that's absolutely changed the outlook for myeloma, probably the biggest advance in the last few years. And this doesn't just obviously include this daratumumab, isatuximab, it includes the other antibodies you heard so nicely about from previously, including elotuzumab. Most importantly, the BITE data are incredibly interesting. Obviously recognizing they're generating very high quality of responses, but the data remain early and we need to be very careful about side effects. AMG 701 may provide much more convenient dosing and hopefully be a little bit safer. The immune therapies are amazing, and the BiTEs and CAR-Ts are obviously doing this, but I really do also have to emphasize the antibody-drug conjugates, because I think, quite honestly, the data around belamaf that Dr. Suzanne Trudell presented were really promising, particularly as we move into combination. CAR-T therapy is great. The true efficacy needs to be determined. I can't tell you when I think CAR-T is going to be approved. Hopefully soon, hopefully this side of Christmas, but we don't know. We'll have to see. Finally, and perhaps in a way most excitingly, is CC220 and drugs like it. These are simply pills once a day, few days on, few days off, and basically combination studies are now underway, and CC480 is also under study. So thank you very much for your kind attention, and close there. Thank you. It's great. It's great. Love you, Sue. Oh, yeah, absolutely, as long as you know.
Okay, so thank you all for bearing with us. Although we're a little bit behind, we had about a half an hour built in to stick around. So we'll do 15 minutes or so, and rest assured, if you asked a question and we don't get to it during the discussion, our nurse navigators back at the MMRF will be in touch with you individually to make sure that your question is answered. So there were topics all over the place, and I just want to get back to a few at the very beginning that I thought were interesting and fair, make sure that they were discussed. One was a simple question around imaging. That was one topic that we didn't discuss here, and I didn't see a lot about it at the meeting, but I might have missed those sessions. So just, help everyone understand what the current guidelines are.
Yeah, absolutely. So you're right, there wasn't too much discussion about it. There was a session about which imaging modalities we should use, MRI or PET-CT scan, low-dose CT scan, because we're using too much PET that may not actually be positive. There was a new imaging modality that Ola Langern presented, but again, that's very early in its development with 2 patients only treated. So I think there is room for improvement, but in general, what we recommend right now is skeletal surveys should be dead, and the future is that better imaging, MRI or a PET-CT scan or low-dose CT scan.
I think it was very interesting to hear some of the newer modalities that are still, I agree with you, very exploratory. We also talked about our PET-CT experiences in different settings, because certainly in the Horizon trial, where we were trying to evaluate extramedullary disease very carefully, we found that PET-CT is definitely a learning curve. We saw literally tumor flare responses, which we haven't seen before necessarily as commonly, and so we're learning a lot. I do agree that there's lots better imaging. I have to say, as an old warhorse myself, I do like the skeletal survey. I use it simply for the purposes of bone mineralization and taking a little look at things, but I do agree with Irene. I'm probably dragging my feet a bit there, but I do like to still use it.
So then there were a few questions around assessing high-risk disease, and I'd even lump into this, although it's a little bit different, the role of genomic sequencing outside of a research setting. Is there a role today in selecting treatment? So maybe start with high risk, and there was a question specifically about amplification of 1Q and whether that's considered high risk.
Yeah. So just remember, FISH and cytogenetics are just methods to look at your genome, but they're big pieces and they're clunky. They're the old-fashioned way. We still do them. It's the standard of care. The hope is that in the future, we will use the new methods, next-generation sequencing, to tell you if you're at high risk or not, because it will be much more sensitive. It's easier to detect the small changes. And it shows us that indeed 1Q amplification, which was neglected before or not seen before, now we're seeing more and more of it, and we understand why it's a bad prognostic feature. Now again, high-risk features all depend, of course, on the treatment, because in the older days we used to say 13q deletion, or we used to say that 414 is bad until we got new drugs that overcome this. So what's bad could potentially be overcome by drugs. But as we know now, there are certain features, something called 17p deletion, p53 mutation, 1q amplification. These are bad players. These tell us that your cells are growing very fast, and we need to overcome them with something more than the current standard of care. And that's why there is so much to be done in immunotherapy, just like Paul said, in CAR-T, in all of those new areas, trying to say, if you have high-risk features when you're starting, or even in the relapse setting, we should definitely think of more options for you than the standard of care.
No, I echo that. I think the important point is about what's actionable. What can you actually change your practice around? And I think that obviously we have the real story of translocation 1114 and venetoclax. What's really true with venetoclax is that if you have 1114 translocation, the clinical benefit from it is extraordinary. It's very real. Similarly, if your tumor expresses high levels of BCL2, even if you're 1114 negative, actually venetoclax may be helpful. So there are a variety of strategies, certainly in the context of clinical trials, but also potentially in the routine practice that can be used. I think the most useful way of doing it is as part of a clinical trial, for sure, because then we know what we're dealing with. I'm talking about whole genome sequencing and the more sophisticated techniques. But I absolutely agree with Irene that the very basics are obviously cytogenetics, FISH, and I must say, I find 1Q helpful. in the sense that it's also typically proteasome inhibitor responsive as a marker, in the sense it's high risk, but a proteasome inhibitor matters when you see it. So there are things that you can act upon. But I would echo a point made by Mary V earlier, that participation in clinical trials is so helpful because it allows that to be collected, and interrogated and viewed prospectively.
And I think my drug is the best example of something where we use your cytogenetics and your information to drive what drugs are. And it always really, it still surprises me. To this date, we understand so much about your biology, about your cells, but then we come back and tell you, well, guess what? We're going to give you the same drugs, RVD for everyone, or their RVD for everyone. Well, one size does not fit all. And the hope is that in the future, one person who has an 11-14 will go on a venetoclax trial or venetoclax therapy because that's where it works. Someone else who has a 17p deletion may go directly into CAR T, and there's a lot to be developed there. And other patients who have 1Q, for example, like you said, Paul, there's the MCL1 inhibitors coming along. So there is more to be done as we develop this, but my drug is the first step forward towards this.
And I just add one other point to that. As we think about whole genome sequencing, we've seen in some of our studies that at diagnosis, the tumor cells, and by the way, when we talk about these mutations, they're not in you, they're in the tumor. They're nothing to do with you. The point is that when you look at the cancer cell by whole genome sequencing, one example, we can see 5,000 mutations in that particular tumor cell at diagnosis. When the disease relapses, in that same patient, we saw 12,000 mutations. So when you're dealing with that, and this just sort of makes sense of what Irene just said, there is a rationale for an umbrella, because how on earth are you going to get your hands around 12,000 mutations? The point is, then you can bring in some specific targets. And I think, and I'm really glad you mentioned that, Irene, one thing I should emphasize with immune therapy is that at least in some measure, it's genetically agnostic, because essentially you're using a component of the immune system to just kill the cell, period, no matter what it's expressing. But we have to be careful about being too simplistic about that, because obviously the cancer has generated the ability to evade the immune system. That must have a genetic basis. And so it's not, some folks have said, antibodies are genetically agnostic. That's really not true. It may be part of the story. But what I want to sort of share with you conceptually is this idea of getting your hands around the whole disease and shutting it down with particular pieces, driving home to eliminate particular clones.
There were a few questions about treating older patients. And in that, I'd say the sub-questions are defining the older patient. Is it just age or other factors? And
sorry, that's all right.
And then getting to the point of, is treatment different or not?
Yeah.
Well, I heard it once from the FDA, and it was really an interesting point. They said we are doing ageism, just like we do sexism and so many things. We are using the age cutoffs, and we should not. So I personally think we should not be using age as a cutoff. As we get older, we all know some of us will keep doing great. I have a patient who runs the marathon faster than me. I know, I always respect that. And there are patients who are frail. So I think instead of using age, we're talking about frailty, meaning are you going to tolerate a stem cell transplant, high-dose chemotherapy, giving you 200 milligrams per meter square melphalan, and really shutting down a lot of the organs, and can you tolerate that or not? And if the answer is yes, then we talk about transplant. If the answer is no, you would not tolerate that because of frailty measurements, then the answer is no, independent of your age.
Yeah, I think and I would take it a little step further. I think the good news is we've developed much more targeted and arguably much less toxic therapies. I agree with you absolutely, Irene. Ageism has no place. I think it does become, are you fit or frail? And there's a concept around fitness that's multi-system. In other words, there are frail patients who may be in their 60s who may not be, for example, great candidates to receive carfilzomib-based therapy because they have hypertension or they have cardiovascular risk of some variety. That I would consider an index of frailty for a particular drug choice. So the good news is, to Irene's point, the old approaches to high-dose therapy have principles that you can apply to targeted therapies. But the good news about targeted therapies is, whereas with melphalan, you're absolutely right, you have to think about everything because it has such an extraordinarily broad effect in its toxicity. I think you can also then think of, with particular drugs, about what particular side effects there may be. The most obvious one, of course, is bortezomib with neuropathy. You may want to avoid that in a person who has underlying neurotoxicity or vulnerability to it, such as someone with diabetic peripheral neuropathy. but at the same time, carfilzomib may not be a good choice in someone who has vasculopathy and would be at risk. The final point I would make is then we have proteasome inhibitors like ixazomib, which are extremely well tolerated. And certainly in my older, frailer population, and by that I mean north of 70 and so on, these are patients in, mid-70s. Again, I'm falling straight down that rabbit hole. The fact is that my point is in frailer patients, ixazomib may be helpful, but I also want to stress I have plenty of younger patients on ixazomib as well, because frankly it's a great oral approach that can maximize quality of life. And I think that's the other point, Anne, if I may, the issue of quality of life. That is so important, and I think that, thank goodness, we saw data on quality of life being brought forward. We're building it into our trials on a routine basis now, and it really matters because it gives us real clues beyond grading of serious side effects as to how a treatment's being tolerated.
And a question in terms of newly diagnosed patients, treating newly diagnosed patients, standard is clearly a triplet, but there are questions on either side. When is 4 drugs appropriate, and is it still appropriate ever just to use 2?
Well, I think 2 drugs up front is very rare, and it would, again, every patient is different, so we cannot say, no, you can never do this. There are some cases, frail patients, who we would say 2 drugs is the right option for you because 3 drugs might be too much. So every case is very different, and that's why I think there is art in myeloma treatment, not just evidence-based medicine. In general, we're trying to achieve the best response as early as possible, and the first treatment is usually what we're trying to achieve it with. So if 3-drug regimen is the standard now, because we know that each of them works in a very different way to attack your cancer cells, the hope is that that 4th drug, especially if it's an antibody that does not cause higher toxicity, will give us potentially a higher response rate. But again, that's not for everyone. We know from the presentation today that even in high-risk cytogenetic patients, that may not be enough, that may not be the right target. And we know that 4 drugs sometimes is not the right answer because they may not give us that much benefit in the overall survival. So there is still a lot to be answered, but we're moving into understanding which combinations are the right ones, whether it's 3 or 4 drugs.
I so agree with that. I think the important point about 3 is probably the base standard, especially for standard risk disease, where you exploit the synergy of an immunomodulator, the synergy of a proteasome inhibitor, and then obviously the WD-40 of the steroids. I think the important point, though, is that we need to think strategically about myeloma, and that actually came out in the meeting and certainly in the debates. We need to think strategically because we've got to think about what we've got to fall back on if treatment fails. And that's very, very important. And I think that was a recurrent theme in the discussions, was how vital that is. So you don't want to be in a situation where everything goes up front, because then if it fails, what's the next move? Having said that, what you don't also want to do is miss the boat if the disease starts to behave particularly badly early. So I think the key thing, isn't it, about the 3 versus 4, is that if there's a clear indication for the 4, sure. If there isn't, 3 is reasonable, but monitor the patient extremely carefully, because then if you need to bring in your 4th agent, you potentially can. And I think that that was a very interesting part of the debates today, was, what goes first and what goes second.
And similarly, focusing on the antibodies that are available or coming, there were a few questions around when to use these and in what sequence. And I'll preface this by saying I know the data's not there, but just your thoughts between daratumumab, elotuzumab, and then isatuximab as well.
Yeah, I mean, that's a hard question. We know that we know that daratumumab and isatuximab are both CD38 antibodies, so they're very similar in the way they act. And it's going to be very hard for us to figure out when to use either or. We know that up front now we're using a lot of daratumumab because of the data. We have clinical data with that. We don't have the data with isatuximab, but we have data in the relapse setting with ISA from what Paul presented. So I think it will be hard for us to put one and then maybe sometime off from a CD38 antibody and then maybe coming back again to CD38 antibodies. Elotuzumab works in a different way. So again, that could be in a relapse setting usually. And again, for certain subsets of patients.
Yes, I think it's fascinating because I think ELO, you see, works primarily through the natural killer cell mechanism. So I like to think of ELO as the alternate to CD38. And the question is, where do you put it? Now, there has been data that shows that when CD38 treatment fails a patient, ELO-based strategies are, of all the options we have, one of the best. There is also data to suggest that once daratumumab or other CD38 targeting therapy fails a patient, carfilzomib with chemotherapy performs well. So there's a lot of important data that's emerging to try and guide us in these choices. I think what's really interesting, though, is that in terms of ISA versus DARA, there are different epitopes, and I think that's qualitatively different because there's more apoptotic activity from isatuximab. What that means is that pathways for killing can be a little bit different, killing the tumor cell, that is, obviously. And in lymphoma, for example, you can revisit Rituxan repeatedly throughout the natural history of the illness. So a patient can receive Rituxan first, at relapse, and then later. I'm not sure that's going to be the case in myeloma. I suspect with DARA, we'll have DARA up front and early. It may be that ISA will be a better choice of CD38 than retreatment with DARA. We don't know. We'll have to see, but it certainly comes to mind. And just very briefly, in my practice, if CD38 failure occurs, obviously to some extent the BCMA target becomes really attractive. And that's why I'm particularly excited, frankly, about belamaf, because it's an off-the-shelf grab for after CD38 failure, where you can just reach for an antibody that does good things to BCMA in the sense of killing the myeloma, and you can just use it. And I'm struck by the DREAM-1 data, where we showed that about 40% of patients responded after DARA failed to that approach.
So I guess a final topic, a couple of questions, of course, around CAR-T, probably a question you get a lot from your patients. Kind of wrapping in, addressing a few points in terms of the timing and process to engineer the cells. I would add one, although it's not here, when would you consider belamaf versus CAR-T? And then how can one get on a waiting list, if you will, for CAR-T?
Yeah, these are hard questions. So I think more, if we look at the BCMA immunotherapy, I would say comparison between CAR-T and the bispecific antibodies, not with belamaf, because belamaf is a slightly different way of action where it's just delivering a toxin. It does have some immune regulation, but it's not truly an immune activator like the bispecifics and the CAR-T. So I think the days when we have both of them approved, it will be an interesting time where we say one is easier and available for everyone, one you have to get the cells out and engineer it and put it back, and maybe that comparison will be a lot easier for us to say, if I have the bispecific now, I'll take it. If not, then definitely I'll work on the engineered CAR-T cells. I think also the CAR-T cells is in the infancy right now. It will get better and better and better. And hopefully we will have it approved so that we're not on waiting lists. We hear it we all have our patients on waiting lists, and we're fighting every single day, keep my patient on, their numbers are good or not. And, it's a struggle. We totally get it. We're all working on this. The hope is that hopefully it gets approved soon and we're not in that struggle anymore.
Yes, I would just add that because of the work we've done with belamaf, that actually there is now emerging evidence to show there is immune activation, and it's through this mechanism by which essentially as the cell dies, antigens are expressed on the surface, activating the T cells around it. So that there is some evidence now, and that's actually also coming from the clinical trials. I think the important point, though, is I agree with Irene completely. They're both different approaches, so they're not exclusively mutually exclusive, is my point. So, for example, in the 916 studies, we have opened eligibility to patients in whom CAR-T has been given. Unfortunately, the CAR-T programs are not doing the same thing. They're saying once you've had 916, you're not eligible for CAR-T. We've made a little bit of a breakthrough here, and one of our sponsors is going to allow a cohort of patients next year to have received prior 916 and be eligible for CAR-T. Because I think the goal here is that we really should have all hands on deck to help our patients. And I think the excitement for me is that belamaf would provide us, say, with something we can use right away, and then be in a position to move forward with CAR-T later if we're having to wait. I do want to say one thing about belamaf. There's been a lot of talk about the corneal toxicity. This has been really well worked out, and it turns out to be very dose and schedule dependent. If you divide doses of belamaf, this becomes much less of an issue. And again, what I'm particularly impressed by is there's going to be a compassionate use program launched in the United States for belamaf, which will not have anything like the rigor of clinical trials to make this drug rapidly available to patients whilst the approval process is underway, because there's nothing more challenging than excellent data in really very ill penta-refractory patients, and you can't get access to the drug until the approval comes through.
Okay. Well, Thank you both. Any final comments before I turn it over to Mary, who will close the program?
Well, we just want to thank you all for being here, for spending your Sunday afternoon with us. And many of you are patients from the New England area or from Dana-Farber. We really want to thank you again for coming. And it's truly a collaboration and an honor to be working with you.
Absolutely. Beautifully put. Thank you very much, everyone.
Thank you so much. And thank you to them.
Great.
Okay, thanks for sticking with us here, everybody. I'm going to just wrap up very quickly. Again, thank you all for coming. Let's give our faculty one more round of applause for their hard work in helping us all learn more about myeloma today. Again, we'd like to thank our sponsors, Adaptive Biotechnologies, Amgen, Celgene, Cure, Karyopharm Therapeutics, Oncopeptides, Sanofi Genzyme, and Takeda Oncology. So don't forget, don't forget, nobody is allowed to take their iPad home. You have to leave them here. But please, while we're wrapping up here today, we would love to have you fill out the evaluation that's on your iPad. There's an evaluation tab, so you can just go to that tab and fill out the form there and then just leave it on your seat. As a reminder, the slides from today's meeting will be available on our website within the next couple of days. And again, the rebroadcast of our live recordings will be on the website as well. So we do invite you to contribute and to attend our patient education programs, our webinars that are held about once a month, and our patient summits. We did have one in Boston back in May, and we're here again today. So you guys have seen us twice this year, so we're getting to be really good friends. So we encourage you to tune in to our webinars and our patient summits. Okay, I just wanted to say a couple of really quick words about the Right Track. And this is a program that our founder, Kathy Giusti, has worked on with a number of other cancer organizations, namely Longevity, the Prostate Cancer Foundation, the Metastatic Breast Cancer Foundation, and the Pancreatic Cancer Foundation. And what they've been able to do is put together a plan to keep cancer patients on the right track toward the best outcome of their disease. And there's really 4 parts to this right track. So the first part of it is to make sure that you find the right team based on your disease, right? So you want to access experts and centers that have a lot of experience in treating the type of cancer that you have. Then you want to make sure you have the right testing done to get the information, the right test, and a precise diagnosis so that you and your care team can make appropriate treatment decisions and then get on that right treatment. It's very important to get the best treatment for your particular cancer, and don't forget to consider going on a clinical trial if it's appropriate for you. And again, you know, the MMRF, we're very big proponents of folks sharing their data at every step. So we always encourage you to do that whenever you can, and certainly through our Cure Cloud initiative, you will be able to do that very shortly. So we have a couple of resources at the MMRF that will enable you to be on the right track with your disease journey. We have our Patient Navigation Center, which is staffed by 3 full-time nurses. Please don't hesitate to shoot them an email or give them a call to ask any questions that you might have, or if you need any resources, you need any information. They're really quite involved with a lot of patients. They're on our Facebook groups as well. They're a great group of ladies, and they really enjoy helping patients. So please don't hesitate to contact them. And also, we have a lot of resources available for you on our website and also on our social media channels. So we do encourage you to be engaged with us in those ways.